Hey everyone, Azim here. We are in chapter 21. We're starting the cardiovascular system. We're going to first talk about blood. We'll also, later in this unit, be talking about the heart and then blood vessels, the three major components of the cardiovascular system. We're going to identify the different cellular elements of blood. We'll describe what that exactly means. Why isn't it just cells? Why is it cellular elements? And the basic functions of these things. We'll review bone marrow and how blood cells are developed. Just a very quick overview. We're not going to go into too much depth about development, just some of the basics. Because these cells are really small, we'll be looking at histology. Histology will be really important for this chapter. Weird metaphor of the day. Blood, the things in your blood are a lot like different things that we find on streets. Blood vessels are like streets, and on streets you find many different vehicles. You find cars, trucks, vans, lots of different things. One type of vehicle you might find is a delivery vehicle. And a delivery vehicle goes from place to place and delivers stuff. If only we got a taco thing to throw tacos at us. But what this is a lot like are erythrocytes. This is a lot like red blood cells. Erythrocytes, red blood cells, they deliver oxygen to all the different tissues in your body. Just like delivery trucks deliver food or packages or whatever, here we have erythrocytes in our blood vessels delivering oxygen. The blood cells don't leave the blood vessels, they just release the oxygen, and the oxygen makes its way to the cells in given tissues. Another type of vehicle we have, set of vehicles that we have, are emergency vehicles. Emergency vehicles like fire trucks, ambulances, police cars, they respond to emergencies of different kinds. What kind of emergencies do we have in our body? We might have an infection. We might have cancer cells. We might have things that our body needs to defend ourselves against. These are just like immune cells. And there's several different types of immune cells that we'll go over. So I'm just saying broadly immune cells. Another word for immune cells are leukocytes. Red blood cells, I should put this in red, but red blood cells are erythrocytes. Immune cells are leukocytes. Oops. Finally, what other cellular elements do we have in blood? They're not actually cells, they're fragments of cells. And what these fragments of cells do, they're just like what these pavers do. What these pavers do, they're patching up parts of the road. Let's say there's a pothole. If you live in a part of town where they actually fill up the potholes, they take asphalt or whatever material and pave over the broken part so that it's a nice smooth ride and there's no further damage. These pavers are a lot like platelets. Platelets patch up any damaged blood vessels. If your blood vessels get damaged in any way, up to a point, it can't help patch up really, really big holes. That requires something further. But if it's a small damaged blood vessel, then platelets can block off and prevent loss of blood. There's more to it than that, but platelets are the first step in preventing the loss of blood. It's the first step in hemostasis, a process you're going to learn more about when you get to physiology. So these are three basic cellular elements of blood. Red blood cells, they're the delivery vehicles. Leukocytes, erythrocytes are the delivery vehicles. Leukocytes are the emergency response. Platelets are the pavers. They repair our blood vessels. We've mentioned blood before. Blood is a connective tissue. Let me remind you, blood is a type of connective tissue. It's not connective tissue proper. It's not one of the solid connective tissues like bone and cartilage. It is a fluid connective tissue. That means it has cells and it has extracellular matrix. It also means that blood came from a specific type of connective tissue. Remember mesenchyme? Mesenchyme led to blood. And mesenchyme came from mesoderm. The extracellular matrix of blood is plasma. It's the fluid extracellular matrix of blood. We've mentioned plasma before as one of the fluid compartments of our body. There's intracellular fluid, there's interstitial fluid, and there's plasma. More than half of our blood, roughly 55% of our blood, is this fluid, plasma. Plasma is predominantly water, about 92% water, and there's other stuff in it too. You've got ions, calcium, sodium, potassium, chloride, lots of other ions. You've got organic molecules like amino acids and proteins and nutrients and wastes. You've got gases, trace elements and vitamins. It's just a whole mixture of stuff. So when you look at plasma, it's just a large mixture of things in your blood. We need fluid in our blood because fluid, water, is a great medium for moving things around. If you have a river, you can just throw something in the river and have it float down the river. That's how, if you're familiar with logging, that's what loggers do. They just throw logs into the river and let it float down to the next site. It's way easier to transport, let the water do the work for you. Here, we can let water do the work for us. Also, the heart pumps the blood. But anyways, you have water just holding the stuff and it can move around in this medium. I'm not going to expect you to memorize every single component here inside of plasma. It's mostly water and there's other stuff in it, nutrients, wastes. It's just, I want you to know that it's a transport vehicle. The plasma itself is a transport vehicle. Besides the plasma, we have cellular elements. We have erythrocytes. I'll use red this time. Erythrocytes, the stem erythro means red blood cells. So erythrocytes, red blood cells. These are the cells that transport around oxygen as well as carbon dioxide. Of that, like if we assume 55% plasma, then 45% cellular elements. Of that 45%, pretty much all of that 45% are erythrocytes. If you spun down your blood using a centrifuge, majority of that cellular element would be erythrocytes. This little bit up here would be plasma. The remainder of it, the 1% left, is made up of two different things. You have different types of leukocytes, your white blood cells, those cells of the immune system. And we're going to go over several different white blood cell types, leukocyte types, that help defend your body against different things. And then the last thing that you can find in your blood, the last cellular element, are platelets. Platelets are not cells, they're fragments of cells. That's why we refer to all these cellular elements, because platelets aren't actually cells. We'll learn how they became fragments in the first place. And platelets are really important for clotting. So these little purpley specks that we're going to see in blood are always there just in case there happens to be a little hole in your blood vessel, it'll immediately patch it up. Let's take a closer look first at where these cells develop in the first place. All of these cellular elements, so erythrocytes, leukocytes, platelets, all of these develop from the same stem cells that are found in the red bone marrow. And as I've mentioned before, these stem cells, they came from mesenchyme originally when we were embryos. When we look at bone marrow, red bone marrow, we've seen this before, but now we're looking more closely. Take a look at this histology. Now, let's look at the illustration first, and then we'll look at the histology. In red bone marrow, you'll have blood vessels. So what I'm highlighting in green right now, this is a blood vessel. Down, blood. You have blood vessels running through the tissue. I also want to mention that the connective tissue that's found in red bone marrow is reticular connective tissue. So you have reticular fibers. And throughout your red bone marrow, found in the epiphyses of long bones and also in flat bones, in between the spongy bone of flat bones. That's where you mostly find it. You have a lot of stem cells. So we have a stem cell here. We have a stem cell here. They're all over, and represented in our histology on the right, pretty much all the cells you see here, save for a few really large ones, these are all stem cells. And blood stem cells can become any kind of erythrocyte or any kind of leukocyte. For example, you can see how, take a look at the left side, how this stem cell could become one type of leukocyte, or this stem cell on the top right can become an erythrocyte. So they could, depending on how they differentiate, they could become any one of these cells. There is a different cell that will lead to platelets. We'll learn that it's this really large one here. I'll just introduce it really quickly right now. These really large cells are called megakaryocytes. Megakaryocyte. There's a megakaryocyte here in this illustration, and fragments of it break off to form platelets. They fragment into the blood vessel. Focusing on erythrocytes first, as I've mentioned, they're a carrier. They're just like a delivery vehicle. They load things up and then deliver things. They're carriers for oxygen gas and carbon dioxide gas. How that happens exactly, you're going to learn that in physio. But what you need to carry oxygen, you need a protein called hemoglobin. Hemoglobin is a protein needed for oxygen transport. If you're at all familiar with hemoglobin, you know that it has iron on it, and iron binds really well to oxygen. So there's hemoglobin found. Let me draw these little dots to represent hemoglobin. Hemoglobin proteins found throughout the cytosol of erythrocytes, and they bind to oxygen. And then think of them as like seats on a bus. They fill up with oxygen, the seats fill up, and then you can go to whatever tissues and let off the oxygen, and the oxygen can be used by cells for cellular respiration, for the mitochondria to use to convert energy. Erythrocytes are shaped, their shape is optimized for oxygen transport. So not only is the cytosol packed with hemoglobin, but you have this interesting shape. It's shaped like a biconcave disc. What that means is, if you look at it from the side, it's kind of smushed in the middle, a little bigger on the edges, almost like you're going to make a full donut, but you didn't push all the way through. So it's really thin in the middle. Thin in the middle means you can transfer oxygen quickly inside and outside of the cell. You have very fast diffusion. Allows for fast diffusion. Additionally, something unique and weird about these erythrocytes is that when they're floating around in your blood, they do not have any organelles. They do not have a nucleus. That's weird because all cells are supposed to have organelles in a nucleus. Otherwise, how are they supposed to survive? Well, the thing is, they don't survive long, relatively. These cells survive maybe 3-4 months before they get recycled. So they have a very short lifespan, relatively. We'll talk about in a minute what happens to them when they die. But by not having organelles, namely mitochondria, they're not going to use up their own supply. They'll take in oxygen onto their hemoglobin, but they won't use it up by any mitochondria because they don't have any mitochondria to use. They don't have a nucleus anymore. That allows them to be thin. That allows them to have that biconcave shape. There's no nucleus plumping up the middle. No organelles means they don't use up oxygen. No nucleus means they have a biconcave shape. Lastly, erythrocytes have a relatively small diameter. The distance across, like take a look at this histology here, the distance across is pretty small. It's one of the smaller cells, not as small as sperm, but it's a pretty small cell. By being really small, it allows them to fit through really small capillaries. Capillaries are really, really tiny blood vessels. If you have really, really tiny branched blood vessels, you're maximizing surface area, you're maximizing diffusion, maximizing exchange of gases, which is the whole point of our circulatory system. Exchange of gases and nutrients. So these erythrocytes, by being really, really small, can squeeze through these tiny capillaries. I should mention here in the histology, take a look at the top left, this histology. The way that this looks under the light microscope, this is a light microscope micrograph. Notice how it's very light in the middle. It's lighter in the center because of that biconcave shape. It's thin in the middle, so light is going to shine through more easily, while the edges are thicker, darker, so not as much light is going through. And you can see they're all pretty much a nice round, small shape. That's the standard shape of an erythrocyte. You look at it under the light microscope. This picture over here would be a scanning electron microscope, which is why you see some three-dimensionality to it. Now, we were just saying how erythrocytes don't have nuclei, don't have organelles. At some point in their development, they had to have nuclei. They had to have organelles. Otherwise, how would you be able to make hemoglobin? Hemoglobin is a protein. You need DNA to have the instructions to make the protein, and then you need the ribosomes and rough endoplasmic reticula and Golgi to all process. They can process the protein. So you at least need that on top of mitochondria and everything else that the cell needs to survive. So early in development, take a look at the top right. Early in development, you have a stem cell here in the red bone marrow. That stem cell differentiates. Oops, excuse me. We have a stem cell here in the red bone marrow. That stem cell differentiates. And you notice it does have a nucleus, it has organelles, but at some point during that development, it releases, it expels a nucleus. So now it no longer has a nucleus, and that's how it gets its biconcave shape. It also allows it to squeeze through into the blood vessel lumen. So here it is inside of the blood vessel now, and there's your red blood cell. At this point, though, it's not, let me back up. At this point, when it has just released its nucleus, it's an immature erythrocyte. The term for an immature erythrocyte is a reticulocyte. You've seen the word reticulo before. Reticulo means network. It has a network of dark staining material in its cytoplasm. Take a look at these reticulocytes at the bottom right. So you don't see it quite has that biconcave shape yet. It will get there. And there's still material inside. Those are organelles that haven't fully disintegrated yet. They haven't fully been broken down. So reticulocytes do get released into the bloodstream. They circulate for a few days before they fully mature into a mature erythrocyte. So it takes some days to form a fully formed functional erythrocyte. Erythrocytes are useful, reticulocytes are a useful indicator. We can measure how many reticulocytes are in our blood to distinguish if we have any problems. For example, if we have low levels of reticulocytes, that would mean that we're not producing much from our stem cells. If we're not producing much from our stem cells, then there must be some problem with our stem cells, with our nutrition. There's an issue with production. Whereas if we have high levels of reticulocytes, that must mean that we're trying to make more than, if we have high levels of erythrocytes and then we would check also our red blood cell count, that could mean that we are trying to compensate for something. We're overproducing unnecessarily, or not really unnecessarily. We're trying to overproduce to compensate for a problem. We're trying to compensate for potential loss of erythrocytes somewhere else. We could be losing blood literally. Our blood cells could be getting destroyed by something else. There's multiple reasons why this could happen. Collectively, what this implies is that we have some form of anemia. The word anemia literally means without blood. An means without, emia means blood. What it really means when we say it is low blood oxygen. And if we have an irregular number of erythrocytes or reticulocytes, then that's an indicator of low blood oxygen. And there's many reasons why we could have low blood oxygen. One potential reason why we could have low blood oxygen is because of a genetic condition called sickle cell disease. Sickle cell disease is where cells look sickled. A sickle is it's a farm tool. It's got a shape like a crescent, like that. And you can see some cells here that have that kind of shape. Sometimes they'll look like a crescent. Sometimes they'll kind of look like a football, like that. Sometimes they'll actually look round, but they'll be darker in the middle. Remember, there's supposed to be light in the middle, not dark in the middle, like that. These are all examples of sickled cells at different angles. They're just because, remember, cells are three-dimensional, so we're looking at different angles. And that's a problem. We don't want misshapen cells. This is a genetic condition. It's a mutation in the DNA that produces hemoglobin. And hemoglobin is supposed to be dissolved in the cytosol, but in this case, you're making a form of hemoglobin that doesn't dissolve, and instead it binds to each other and it crystallizes. That's what I mean by crystallization. Take a look down here. Here's normal hemoglobin, all nicely dissolved, versus here where abnormal hemoglobin is forming long, jagged crystals that stretch out the shape of the cell. That gets caught in blood vessels, that gets destroyed by our spleen and recycled at too high a rate, and that can be problematic for our spleen. Lots of problems can arise from this. So this is one example of anemia. It's a genetic disorder. So you have to inherit it from your parents. Another form of anemia, and I want to say that there's like 90 plus types of anemia. I'm only going to go over 3. One type of anemia is sickle cell. Another type of anemia is macrocytic hypochromic. Macro means big cell, macrocytic. Hypo means small or low. Chromic means color. So low color means pale. Big cell that's pale. Take a look at these cells here. They're larger than normal. The paleness is a bigger spot than normal. So it's a big, lighter colored middle. Cells become this way if we lack vitamin B12. Vitamin B12 is an important vitamin for DNA synthesis or replication, one of those. Has to do with mitosis. So if you lack this vitamin, if you are deficient in it, then you might end up getting macrocytic hypochromic. So this isn't genetic, this is environmental. Whether or not you get the right amount of vitamin can lead to this condition. And this is a problem. Larger than normal cells won't work properly. They'll get destroyed by our spleen, and other problems can happen. The third type of anemia that I'd like you to know is microcytic hypochromic. Microcytic hypochromic, it's a smaller than normal cell, and it's still pale. So take a look here, smaller than normal cell. Look at how big the white is relative to the red. They're smaller than normal and pale. I know they look a lot like regular erythrocytes, but they are smaller than normal. And look at the white space. The white space is pretty big, so that edge is thinner. When it comes time for the mini or practical, I'll give you a side-by-side pictures to compare, and you can pick which one you think is which. This one is called by iron deficiency. If you lack iron in your diet, you can get iron from red meat, from green leafy vegetables, from, I'm sure, other things, but those are two things I can think of right now. How are you going to remember this? What they're caused by? Do you know Mike Tyson, that boxer? He was known as Iron Mike. Iron Mike. Microcytic hypochromic anemia is caused by iron deficiency. And then vitamin B12 leads to macrocytic. B enlarged, that's big Mac. That's another way you can remember it. And then sickle cell, it's genetic. I don't have a specific mnemonic for that. Sorry. Hopefully you can let me know of one. Right, so those are the three types of anemia and what reticulocytes are. Reticulocytes are immature red blood cells, and then erythrocytes as a whole, what they do and why they're shaped the way they are. That's the first part. Next part, we'll go over leukocytes and platelets. Thanks for watching. Let me know if you have any questions. I'll see you in the next video.